Hi, today I will be presenting on the stimulation of gastric acid secretion by rapid parietal cell A2B adenosine receptor activation. So parietal cells are found in the gastrointestinal tract and they are responsible for secreting acid into the lumen, and in humans they are also responsible for secreting intrinsic factors. These parietal cells have apical and basolateral transporter proteins that secrete ions such as H+, K+, and Cl-. Gastric acid secretion is associated with three specialized cells: chief cells that produce pepsinogen, enterochromaffin-like cells that produce histamine, and G cells that secrete gastrin and promote acid secretion. However, gastric acid secretion is inhibited by somatostatin. The most important stimulus of gastric acid secretion involves the histamine H2 receptor. Gastrin binds to its specific receptors on the surface of ECL cells in order to induce production of histamine, which serve as ligands for the histamine H2 receptors. The histamine receptors activate adenylate cyclase in the parietal cell, which increases the concentration of cyclic AMP. The increased cyclic AMP levels results in active protein kinase A phosphorylating effector proteins related to the transport of the H plus K plus ATPase pump from the tubular vesicles in the cytoplasm to the cellular membrane. These pumps secrete H plus ions out while untaking a K plus ion in. This pump requires energy from ATP in order to secrete these protons out of the cell. While protons are being secreted out, two additional things must happen. The first one is Cl ions must be secreted out of the cell into the lumen in order to bond with the protons to produce HCl. The other involves the recycling of K plus ions so that there is an optimal concentration of K plus ions in the lumen to be used in the H plus K plus ATPase pump. Adenosine is a purine nucleoside, which is a nucleotide without a phosphate group. Adenosine modulates chloride secretion in the gastric glands. Extracellular adenosine molecules bind to four different receptors on the cellular membrane: A1, A2A, A2B, and A3. The A2B receptor has the lowest affinity for adenosine, although recently research has gained momentum on this receptor for its potential use in cancer treatment and as an anti-inflammatory. When there is an inadequate supply of blood to a part of the body, also known as ischemia, the adenosine levels rise to activate the A2B receptor sufficiently. When oxygen level is low, the A2B receptor is overexpressed. The overexpression is due to an HIF1 factor binding to the promoter of the gene that encodes the A2B receptor, thus inducing overexpression. Recently, it was found that this gene was expressed in mRNA and protein levels in rabbit gastric parietal cells. These A2B receptors have also been confirmed to be present on the cell surface by flow cytometry and confocal microscopy. A2B receptors seem to be co-localized with the adenosine deaminase, an enzyme that converts adenosine into ionosine. This close interaction facilitates the ligand binding of adenosine to the receptor to activate the GS protein for further intracellular response. It is suspected that adenosine may serve a role in regulation of the secretion of gastric acid by binding to the A2B receptor in the parietal cell. Parietal cells were isolated from male and female New Zealand rabbits. These parietal cells' functionality were determined and observed by measuring the hydrochloric secretion at a concentration of 10 to the negative 7 molar to 10 to the negative 3 molar histamine. The sample of these parietal cells was assumed to represent a normal population of resting parietal cells. Acid formation was measured using the aminopyrin accumulation method. Parietal cell membranes were isolated using 20 millimolar tris HCl buffer and 50 millimolar tris HCl buffer. The cell membranes were then resuspended in the 50 millimolar tris HCl buffer and stored in negative 80 degrees for usage. The protein concentrations were determined using a Bradford assay with BSA as a standard. Ligand binding experiments were carried out using NECA for the competition binding experiments. The A1 receptor agonist, A2A agonist, and A3 receptor agonist were used for the association, and DPCPX, ZM241385, and IB-MECA were displacement agents in the association/dissociation kinetics experiments. Adenylate cyclase activity was measured by quantifying the amount of cyclic AMP generated. AP accumulation was determined to represent the H plus produced and compared it to the increase generated by the histamine ligand. As seen in Figure 2a, the AP accumulation was determined when adding histamine. Using increasing concentrations of 2-CADO, which is 2-chloroadenosine, an adenosine agonist, the AP accumulation was determined, also seen in Figure 2b. The AP accumulation from 2-CADO was not very different from that produced by histamine, so it was chosen to be used for further studies. To test whether the stimulation of acid secretion by 2-CADO and the A2B receptor is mediated by cyclic AMP levels within the cells, IBMX was applied in specific doses. IBMX is an inhibitor of the cyclic AMP phosphodiesterase, so adding this inhibitor would result in amplified responses from the parietal cells, meaning a higher AP accumulation. This means that with the A2B receptor activation by the 2-CADO, we should see amplified effects in the AP accumulation as the cyclic AMP will continue to activate protein kinase A to phosphorylate the target effector proteins. As seen in Figure 2d, the addition of IBMX substantially increased the AP accumulation in response to 2-CADO. A similar effect was seen in Figure 2c as the addition of IBMX substantially increased the AP accumulation when histamine was used as a ligand for the histamine receptor. Both of these responses were measured with two different concentrations of the ligand, the first at 10 to the negative 6th molar and the second at 10 to the negative 3rd molar for the histamine. For the 2-CADO, the two concentrations were 10 to the negative 5th molar and 10 to the negative 3 molar. These results showed that the 2-CADO acted in a coupling manner with either the A2A or A2B receptors due to the effects seen when there is an increase in cyclic AMP levels. In order to determine which receptor is involved in stimulating the acid secretion, antagonists for the other subtypes of receptors were used to see if there would be an effect on the AP accumulation. This method of determining which receptor the ligand binds to is called the agonist and antagonist binding assays. DPCPX at a concentration of 10 to the negative 7 molars was used as a first antagonizer. DPCPX is an antagonist to the A2B receptor, and adding this antagonist at this specific concentration inhibited the 2-CADO response as there was a significant decrease in AP accumulation as seen in Figure 3a. When this was compared to Figure 2b without the DPCPX, you could see that there was a significant decrease in the AP accumulation. SCH58261 at a concentration of 10 to the negative 9 molar was used as a second reactant. The SCH58261 is an antagonist to the A2A receptor, and adding this antagonist to the 2-CADO reaction showed virtually no change in the AP accumulation as seen in Figure 3b. This means that the response generated by the 2-CADO application was independent of the SCH58261. DPCPX at a concentration of 10 to the negative 9 molar was used as the last antagonizer. DPCPX at this specific concentration serves as an antagonist to the A1 receptor. DPCPX, however, at the previous concentration of 10 to the negative 7 molar is an antagonizer for the A2B receptor. When the DPCPX is applied at a concentration of 10 to the negative 9 molar, it serves as an antagonist to the A1 receptor. And with the addition of this antagonist, there was virtually no change in the AP accumulation as seen in Figure 3d. These results indicate that adenosine and its derivatives may stimulate acid formation. Statistical analyses were performed to compare the two groups. T-tests were done in order to test for the difference between the AP accumulation between two groups with the inhibitor and without the inhibitor. The null hypothesis was that the two values would not differ, and since the p-values were calculated and came out to be less than 0.05, we were able to reject the null hypothesis that they do not differ, meaning that there is a very good chance that they do differ. 1 and 2A ANOVA tests were used to determine whether there was any significant difference between the two groups as well. In conclusion, the stimulation of acid secretion by the 2-CADO and the A2B receptor was found to be mediated by cyclic AMP levels within the cells. The A2B receptor was also determined to be the receptor involved in stimulating acid secretion, not A2A. So, all these results combined indicate that adenosine and its related derivatives stimulate acid formation and secretion in rabbit parietal cells through A2B receptor activation.